Hello, everyone, and thank you for attending today's webinar, Unlocking Efficiency, Strategies for Expedited Biotherapeutic Development, presented by Bionova Scientific. I'm Rebecca Kushmiter, and I'll be moderating this webinar. I'd like to start by introducing today's speakers. We'll be joined by Minnie Ashwath, Senior Director of Process Development at Bionova Scientific, and Daniel Bach, Senior Director of Upstream Process Development at Bionova Scientific. You can read their full bios on the left side of your window by selecting the Speakers tab. Just a few quick notes before we begin. You can access closed captions from the bottom right corner of the video player. The webinar is being recorded. It will be available to watch on demand within 24 hours. And we'd love to hear from you. During the presentation, please submit any questions you have using the Questions and Answers tab on the left side of your screen. Time permitting, we will conclude with a Q&A session. Okay, now I'd like to turn it over to our speakers. Minnie and Daniel, please go ahead.
Thank you and welcome to the talk. Today we are hoping to present a very high-level overview of drug development and some strategies how we can expedite some of the essential activities. Just as an agenda, we will introduce briefly about our company, Bionova Scientific, and our parent company, Asahi Kasei. We will give an overview of drug development starting from a clone all the way to commercialization. We will outline some strategies for successful path to clinic and things to avoid for any kind of obstacles. And we will also provide some strategies for a successful path to commercialization.
Yeah, good morning also from my side. I would like to start with the introduction of Bionova Scientific. So Bionova Scientific is the full biologics CDMO, and we start our service with cell line development, process development, up to providing GMP material from our manufacturing facility. We were founded in 2014 in the San Francisco Bay Area, and we have a highly experienced teams of scientists and engineers. We have scientists and engineers from big pharmaceutical company, but also from small startups and everything in between. And in 2022, we were acquired by Asahi Kasei, and since then we are part of the Asahi Kasei family. And here I would like to give you a quick picture how it looks like. So Asahi Kasei has all kinds of business units, for example, chemicals and electronics, but also homes, construction materials, pharma, medical, and also healthcare, to name only a couple of them. What you can see here on the slide are very popular in the industry, the Planova and the BioOptimal filter, that are filters that are usually used in purification processes. But we have also fluid management systems like chromatography skids and also virus filtration skids in our portfolio. We have some sister companies, for example, VirusSure is one of our sister company. VirusSure is in virus safety. For example, they are doing virus clearance study, or BioUnique, they are specialized in mycoplasma testing from raw material up to cell culture or downstream intermediate pools. And then, of course, as I mentioned already, Bionova is also part of the network. We provide full service development for biopharmaceutical processes and then manufacturing in our GMP area.
Any of the biologics development really go through three distinct stages in the life cycle. We always have the process in mind and the product in mind. With the end goal, we start with the process design, where we design an upstream, downstream, and analytical method development that best suit the product. And you have, from a CMC perspective, these are required to ensure safety and efficacy for the product. And as the product moves through these stages, you have the basis of knowledge management, and you pay attention to the regulatory guidelines, and you are working with different teams. It's a very cross-functional effort, and you have to focus on criticality of the product quality. And the focus is on risk management, and the product moves through these stages with high efficiency, and there are places where you can cut down the timeline based on risk assessment. So this presents an overall drug development timeline. This is a very traditional approach. Here we are presenting the different phases, right from the kickoff of a project, from cell line development, where you have identified a stable clone, and we have divided our development approach into multiple phases. To support tox studies or phase one clinical supply, we are calling that CMC1, Chemistry, Manufacturing, and Controls, and this is stage one, where we are developing a clinically ready product. Here, the focus is speed to clinic, and the development is focused on yield, titer, what have you, but it's really rapid. And this is where you can save some of the time to get into the clinic, and you want to get the first manufacturing run completed so that you can get into the phase one stage. And after that, you have your CMC2 development, where you're starting to focus on the commercial development and also moving the clinical studies into phase two and phase three. And here is when you start looking at the process optimization, robustness, which we will get into a little later in the talk. But you are really trying to develop the robustness and the deep understanding of the process, which eventually lead to PPQ, which is the process performance qualification, which are required at scale studies to be able to file your BLA document, which is the biologics license application. And as the molecule moves, we are keeping those three validation stages that I outlined in the previous slide, and through risk mitigation, risk management, we are moving these molecules as fast as possible toward launch because patients are waiting. Throughout this general development, there are some strategies that can be applied. I will not go too much into detail about these strategies because they're heavily molecule dependent. But there are four pillars that we follow here at BioNova. The risk-based development strategy. We have strategy-based end in goal to develop this life cycle approach. We have well-established cross-functional teams which work in cohesion to move this molecule along. And all this is managed by good documentation processes and practices. Knowledge management is key in CMC and good CMC development. And we can leverage a lot of information that's published by either the international harmonization guidelines, the ICH guidelines, or by the regulatory agencies such as FDA or EMA. At every step of the process, there are guidelines to industry, and these are again guidelines, and they can be adapted to your molecule. And when you follow these guidelines, acceleration is possible, justification is possible. In in Bionova, we are working with various modalities, not just regular monoclonal antibodies. So we have our platform, which is based on FC-containing molecules, where the development is rapid. We have a really well-formed platform where you can plug and play these molecules. So when we get a clone, we have a platform for upstream cell culture production. We have a platform for all the purification unit operations. We have platform high throughput methods to analyze these samples. And so when you have a platform molecule containing FC region, you have shorter development timeline. But we're not skittish about dealing with non-FC modalities. We have experience working with enzymes, tough proteins, single chain FVs, nanobodies. And so we have a toolbox. Instead of a platform, now we rely on our toolbox to be able to rapidly move the molecule through, especially the clinical stage, which is a CMC1. And our end goal is to have a robust process that can be manufactured in our manufacturing facility, which is also in-house. So all this approach starts with the platform fit assessment. We optimize to meet quality targets at every single stage, and we do the process lock. The overall objective, as I mentioned, for CMC1 is speed to clinic, but we are customizing CMC solutions to meet the life cycle and end goal. We start with molecule evaluation and gap analysis if it's hydrophobicity is an issue, or if it is a low titer, if that's what we need to focus on, and optimization to meet product quality targets, whether we need to run gradient dilutions or what have you. And of course, the end goal is to fit it into our facility. We have 500-liter, 1K, or a 2K GMP manufacturing facility. So our end goal is to be able to seamlessly transfer to our facility. We have to identify quality target product profiles, the QTTPs, at this stage to enable rapid transfer of these processes.
Okay, so as Minnie already introduced a couple of slides ago, that's the drug development overall timeline. But in the next couple of slides, we would like to focus on the early stage development. And early stage development encompasses cell line development first, and then followed by CMC1 development. Also, material supply for tox studies, and then followed by a GMP campaign for clinical phase 1. So this is an example how it could look like for upstream early stage process development. Usually we start with a high throughput screening at the beginning, and in this high throughput screening, we test many different factors on 2 different levels, because here we would like to identify the factors that had the biggest impact on the cell culture performance as well as on the product quality. Sometimes we do a final clone selection in this stage, that we have the top 4 clones in combination with different medias, but sometimes we also look already if we get the final clone into process conditions like temperature, agitation, dissolved oxygen, and pH. So after the high throughput screening, we go into an optimization round where we test then less factors on multiple levels. That is really the step where we fine-tune the process. And usually in this step, we have the final clone selected, we have the media selected. And again, so here we are fine-tuning the concentration of a component that it's added, or a pH, a temperature range, a temperature shift, things like that. So after the optimization, after we identified the best producing process with the best product quality profile, we scale it to our pilot plant facility, and there we execute the process in a 50-liter or 200-liter single-use bioreactor. This gives us then also the opportunity for a harvest study, and this material can also be used for tox studies. So in our pilot plant, we have the same equipment like in our manufacturing facility, of course, a little bit smaller. So that makes sure that we have a smooth transfer from our pilot plant then later to our GMP facility.
On the downstream side, development really starts with DOE-based screening, whether we apply high throughput methods, or we used something like a high screen column. The focus here is to have limited screening of capture step. It's something that you could flow through really fast. We want to get to those polishing steps that really make the quality and the purity of our targets. So our capture step and the virus inactivation steps that are typical for biologics are very quick, and we try to use membrane-based technologies as much as possible, mainly because of the single-use aspect and also the higher throughput aspect. And we have targeted screening for polishing steps. And this could take anywhere from 1 to 2 months, again, expedited by the platformability of the molecule. For antibodies, definitely we have a lot of information either through experience or through literature, whether it's bispecifics, they are posing little greater challenges, but we have a platform for those as well. And as upstream is scaling up to lab scale runs, say 2-liter runs, we are supporting the development in parallel. And these are limited runs to confirm findings using the DOE screening. And of course, the material is getting more and more representative as upstream is locking their process. We could use this stage for material supply for various studies, especially if we are doing formulation development, which is an activity that requires quite a bit of material. And we can supply material for analytical method development or any kind of testing activities. Here we are moving slowly toward the bed height that is going to be finally transferred to our GMP facility as well as our pilot plant runs. So we are using lab scale columns. And the time here can also be expedited by prior knowledge. And if we already have those QTPPs identified, like I mentioned in the previous slides. And as upstream is scaling up, they are generating material more on the pilot scale runs. We have capability to run up to 200 liters, as Daniel mentioned. And this is really an essential activity that is required to transfer a process, especially downstream, where you would have developed the process in bits and pieces. This is really the first time we're putting the whole process together end to end, all of the unit operations from capture all the way to final filtration of the drug substance. There are additional tests that are conducted here that support the GMP manufacturing, like hold times and freeze thaw and filter throughput, etc. More critically, we are supplying material for analytical method confirmation and additional formulation development. There is the concept of a process lock from this stage. So the findings we have from this pilot scale run is what goes into a process description that get transferred to our manufacturing facility. And we can also produce tox grade material from these pilot scale runs. And as Daniel mentioned, all of our downstream pilot scale equipment mirror what we have in the manufacturing suites. And an added advantage to that is now the manufacturing folks who are the operators for these molecules can come and already gain an experience in our pilot plant without having to gown in and even before the molecule goes into manufacturing.
Okay, yeah, we talked already a little bit about transferring the process then into our manufacturing facility to produce clinical material. In this process, we call it NPI, and new product introduction. And here you can see the overall timeline. So on the top of the timeline, you can still see the upstream confirmation run, what comes first, of course, then followed by the downstream. And then in parallel, we already start working on the documentation, like the process description and writing the development report. And in parallel, our colleagues from the MSAT team start working on the new product introduction to get our manufacturing facility ready to produce in the process under GMP condition. So here it's really important that these activities happen in parallel to shrinken the timeline. So Mini and me, we are still working on the development report NPD, and manufacturing is already producing batch record and the at-scale process description. In parallel, also the planning BOM is ongoing. You see here on that chart only the final BOM. The final BOM is approximately locked by the thaw in manufacturing, but before that, we are working on the planning BOM. So as soon as we can lock a material, it gets locked into the planning BOM that our colleagues from supply chain can start ordering the material. And as I said, so this planning BOM will then be finalized by the time of thaw. Of course, all the materials that are needed for the thaw are locked already a little bit earlier. So, and then, of course, all the activities in process development in our MSAT team will result then in a thaw in manufacturing. And approximately 1 month after all the activities in process development are completed, we do the thaw in our manufacturing facility.
And once manufacturing is done for phase one supply, it could be 1 or 2 or 3 batches, depending on the clinical need. There are multiple other activities that start. Of course, you have the stability program, you have a little more extended characterization that happens. But most importantly, now the program is moving toward the regulatory filing. And this is where in my observation and in industry reports, there have been a lot of delays or rejection of the IND itself. And I have outlined 4 very high level strategies where we can avoid delays in the IND submission, and also the worst can be the clinical hold. The last thing we want to do after all these few years of development, we don't want to go into a clinical hold. Number one is communicate with the FDA. I'm only using FDA as an example, but it applies to the European Medicine Agency or any other health authority where you plan to do the clinical trials. FDA has a special program called the Interact program, where you can um, engage in early discussions with them, especially for biologics. Meetings prior to IND submission really help build that the confidence with the regulators, as well as give tips and tricks for the IND submission. Of course, the IND should be well written and error free. There are multiple modules. I won't go into too much of the details, but each of these modules has a purpose. And a lot of our CMC sections are outlining the manufacturing process. What is the identity? How are we proving identity? How are we proving that it is safe? So there are very strict guidelines on these written in the Common Technical Document, the CTD. The third important point to avoid delays is the detailed description of the manufacturing and other CMC requirements. I already mentioned Module 3, but within that, there are multiple sections. They could range everything from describing the upstream process to the analytical methods to the virus clearance studies, the 2-virus clearance study that is required at this stage. So the data have to be consistent and complete. And there has to be the strategic planning for regulatory compliance. And that is why we have to have the life cycle approach in mind. It is just not the IND submission. We're not stopping here. Our end goal is entry to market, where we actually help patients on a larger scale. So that strategic regulatory compliance mindset has to start with the IND submission, because we can use many of these documents and leverage them into our BLA filing to prove continuity. Consulting with regulatory experts to speed up, and they are aware of the latest guidelines that the agency may be putting out. And here at BioNova, we are monitoring those updates, especially with the FDA around these topics.
So after the early stage development, of course, you will have the late stage development. And in late stage development, you want to further optimize the process. This is also the phase where new material is supplied for additional clinical studies. And at the beginning of a late stage development or also CMC2 development, it's not uncommon in the industry that you have a tech transfer between sites or also tech transfer between different CMOs. So for a tech transfer, it's really important that you understand in depth your sending facility and your receiving facility. So because the first step is to do a gap assessment between both sites to really identify the gaps. And after you identified the gaps, you can, you know the risk. What's the risk for that tech transfer? Knowing the risk, you can create risk mitigation studies, and with that risk mitigation studies, you can reduce the risk. One example could be the bioreactor geometry could be different. For example, the spar jar. So with different spar jar, with different pore size on the spar jar, you can get different size of bubbles in the bioreactor, and you see it here in a picture on the right. So basically, with smaller bubbles, you rupture the cells on the surface of the liquid, and with that, the viability drops. You definitely want to avoid that to see that in manufacturing. That is why you can design risk mitigation studies earlier in the process development stage. Other example is that maybe a vendor is qualified on the sending side, but not qualified on the receiving side. But there are alternatives, of course, available on the market. And there you can show with studies that raw material from a different vendors results in the same process performance. As I said, the CMC2 development, it's also important, or we optimize the process there further and make sure that it really fits into the commercial facility. So again, it is process optimization, you want to get a higher titer with a product quality to match your CMC1 development. You look into the raw material again, and of course, you try to reduce the cost. To speed up the overall process for the drug development, it's really important to look into this stage already into the robustness of the process. So you should identify critical quality attributes, and we need to understand or getting to know our key and critical process parameters. So in this phase, we establish ranges for the next manufacturing campaign. And also it's important really to get here a strong data set, really in-depth knowledge about your process. That helps speeding up the following steps. Yeah, the following step is then pretty much getting ready for a process characterization study. And here the most important thing, or a very important thing in the preparation for the PCS, is that you have your knowledge summarized. So you have CMC1 development, you have CMC2 development. As I said, sometimes you have tech transfer between different sites or different CMOs. And different manufacturing campaigns, maybe in different scales. So you need to summarize all this knowledge and make sure that you have this knowledge available in one location. Of course, you need to understand your input and output parameters and the relationship between it. And then you start further doing the preparation for the FMEA workshop. So that means, for example, equipment capabilities understanding. That helps then later to design actually your experiments. Then we will have the FMEA workshop where we look in all upstream and downstream parameters, and that are probably around 800. And the FMEA is pretty much a prioritization of all these parameters to identify the parameter that really impacts later your product quality. You rank them based on severity, occurrence, and detectability. And this is a cross-functional workshop from people from process development, from our MSAT colleagues, from our engineering department, and also from our quality department. And then, of course, before you can start the actual PCS, you need to work on the documentation. You need to finalize the FMEA. You need to have a process characterization master plan in place. And then, of course, you need to have a protocol for every individual study in place. So this is an example for upstream how a PCS could look like. That is a work package. So I already explained the FMEA. So if there was no scale down model developed before the process characterization study, this is a place where we need to develop a scale down model to make sure that you can show in small scale what later will happen in your manufacturing facility. After the scale-down model development and the scale-down model qualification, you go into the actual PCS, what is then a big block of DOE experiments. So for upstream, that are probably 50 to 100 small-scale experiments. It depends on the outcome of the FMEA, of course, and that will be designed by design of experiment and then executed in several blocks. You will probably have 3 to 5 different blocks with bioreactors around 20 to 25. So then usually a media stability study is executed during that step. So maybe there's commercial media available, then the data are already available, but there are also customized media on the market. So if there are no data, how the storage condition and the shelf life impact the media stability, then that is a place where you need to investigate that. N-1 linkage study is where you look into everything, what happens in the pre-stage bioreactor, how does it impact the end-stage bioreactor or the production bioreactor. For example, pre-stage bioreactor can grow for a day longer because the production bioreactor is not ready yet. That is what you would like to investigate, how then this 1 day longer cell growth impacts the production bioreactor performance. Kind of risk mitigation is a small-scale limit of in vitro cell edge study. To execute that during the process characterization study, it's not required by the authorities. By the authorities, it's required for the commercial scale. But you don't want to do that. You need to do that during the PPQ, but in case there are any stability issues, you want to find that out before the PPQ to address these kind of issues before. And then, of course, a PCS is also the place where you need to look into your harvest process in case the harvest process was identified or scored with a higher risk.
As we're moving from this early stage to late stage to commercial readiness, what does it mean for CMC? That is the next stage of the drug development process. So, to recap, up until now, we have a viable upstream downstream process. We have methods to be able to identify the impurities, both process related and product related impurities. We have all of the regulatory documents in place to be able to move the molecule from clinical to commercial. And this is the focus in this commercial readiness piece. Now, in addition to the process, we are also focusing on the facility. A lot of focus is given to the quality systems, the way we are addressing deviations and all of the quality checks that come in that are required for commercial production. And this activity and the planning will take very diligent cross-functional team to work together. So as we're getting ready for commercialization, we mentioned briefly about the PPQ. PPQs are process performance qualification. These are at scale experiments, if you may. So in all of the stage one, the PCS that Daniel described, we are doing this in a non-GMP facility in small scale, but bound by statistics and the QA agreements. And now, when you're doing PPQ, these are done in the large scale, at the commercial scale that is decided. So, a lot of times, we have to we have to know what the commercial scale will be. And that is the knowledge that will help expedite the process. In stage one validation, these are again can be done in parallel to PPQ to expedite the timeline, or it can be done along with PCS. It just depends on the strategy, the life cycle strategy. For downstream, in addition to all of the process characterization studies where every unit operation is studied, there are a few other required studies to be filed into the BLA as regulatory requirements. Scale down model qualification is a very important activity. How can we show what we did in small scale can uh, can can be the equivalent in a larger scale in a column, for example. When we scale up, we have to have that confidence that the impurity clearance or the product quality targets are met regardless of scale. The 4 virus clearance studies become important. Now we are really getting into the adventitious agent safety studies. And the resin and the membrane lifetime studies become important as well, where you have to prove to ourselves and to the agency how long can you use a particular membrane or a particular resin. The more, the better, of course, but we have to demonstrate cleanability or consistency and robustness of the process. Whole time studies are also studied in small scale. And some of these you see on the right hand column are mirroring where we have to repeat those studies at scale because it has been proven that there are certain studies that cannot be inferred, the scalability cannot be inferred in smaller scale. So you will see the studies on the left are done in a non-GMP area. They have to be repeated through a validation protocol on larger scale on the GMP runs during PPQ. The PRI assessment, some of these can be done as paper exercises. They don't really require experimental setup where you are assessing product related impurities. Some of those you may have to test based on the severity that is recognized. We assess refiltration, whether it's from the refiltering of the virus filter or of the final drug substance. and those have to be validated as well. One of the major contaminants that is an immunogenicity risk is DNA that comes from the CHO cells itself. And in downstream, we have to prove that an excessive in an instance where excessive DNA comes through, our product process can be able to clear them. Critical raw materials are identified and evaluated, and extractable and leachables are assessed because there's a lot of use of plastic in the process. So after completing all these studies, now we are really getting closer to the BLA. As a summary of everything we have said so far, we have how do we know we're ready for late stage? How do we know we're ready for PPQ? There are very clear instructions. And as a site, Bionova, we have developed these processes in place, these stage gate approach, where we each stage gate signifies and signals to the teams, hey, we are done with this stage. It's moving on to the next stage. So we know we are ready for late stage when we have the commercial process locked through CMC2 development. We know we are ready for the PPQ runs when we have enough clinical experience and we have that confidence to establish the critical quality attributes. And the ultimate goal is to come up with a control strategy, and that is the requirement for BLA submission as well. Through this control strategy, we are saying we're ready to manufacture, we're ready to go on to the BLA, and all of these activities are statistics based. A few strategies to help avoid delays in BLA filing and commercialization. This is a huge activity that has happened up until now. It takes about 12 to 16 months to complete process characterization. It takes about another 12 months to complete the PPQ and release those batches. And it takes another 6 to 12 months to complete the BLA filing. So as you can see, cross-functional groups, lots of different hours spent on these, and the BLA filing delays can be disastrous to the company, both in terms of revenue as well as market capture. Every month there is a delay, there is a danger that there is another company coming into the market and you may lose your opportunity. Again, communication is number one, just like how I outlined for the IND submission. Communication with the agency may at this time it could be multiple agencies. Many drugs are targeted to international markets, whether you have the FDA, EMA, or Health Canada or Brazil or Japan. These are some of the major health organizations. So a lot of the drug development strategies are based to cover these multiple clinical markets. And it's critical because many of the oncology targets the world it's facing, and you want to be able to launch a product to help the patients in all these health agency areas. The as as in an IND, the studies and the documentation have to be very comprehensive. The agencies are really interested to know what you did not test more than what you actually tested. That justification for not testing a parameter in PCS or not testing in PPQ have to be clearly outlined. And you have to demonstrate that you have put adequate controls in your manufacturing to be able to reproducibly and produce these high quality in the commercial scale. The robust control strategy is one way that ensures safety and reproducibility of these batches. That is where all of the small scale studies, the non-GMP studies, the development runs, the clinical runs, they kind of all feed into this ocean, the control strategy. And each manufacturing site has to come up with their own control strategy. And Daniel mentioned in one of his slides how there are transfers between sites. So, and we are specialists in those kind of transfers. We can quickly assess facility fit. We can look at the gaps that are there, having the BLA in mind. What are the studies that we need to run to ensure adequate control strategy for commercial production? One of the misses some companies may have is to have a very weak continuous process verification strategy, which is post-market, post-commercialization. You have to have a very robust CPV program. If you don't, it's a miss, and it could help. It will contribute to delays in your BLA verification or BLA approval. PAI, the pre-approval inspection, is a very critical activity that can also impact the BLA timeline. This is where the agencies come to your facility, and there is a big audit to make sure that you are truly ready to produce these commercial drugs that go into patients. A well-defined post-market strategy is, I already mentioned, the CPV program, and it's not just the CPV. The sponsors have to have a pharmacovigilance groups, multiple vigilance groups, and you're always looking at the outreach of your drug. So, but from a CMC perspective, having that CPV, having reproducible data, filing those data with the agencies on time, those are the commitments you make in your BLA filing. And if any of those are missed, that lead to the delays. So in the last 40 minutes, Daniel and I hope that we have given you a very basic, very broad overview of the CMC pathways. To conclude, there are some points we'd like you to take back to your daily activities. Careful planning is needed to navigate these CMC activities. Always have the end goal in mind. Whether you have accelerated programs, breakthrough or orphan designations, BioNova can work with you, and we incorporate all of those philosophies in our development process. As I mentioned earlier, and Daniel mentioned as well, we are adept with working with small or large biotech or pharma companies. We know how the processes move, so we can truly advise on how to avoid delays, expedite, whether it's development, expedite manufacturing. We specialize in developing and manufacturing various modalities. So now is the time in the industry where we have multiple modalities, and it's not just the monoclonals, and they pose several CMC challenges. We provide customized, innovative solutions to address CMC strategy. If you say, why BioNova? Why should you choose BioNova as your CDMO? I could say that in the last bullet. Our scientists and engineers are highly qualified, and we can support throughout the life cycle of the program, whether you come to us with your gene of interest or a commercial process and everything in between. So Daniel and I would like to thank you for listening to us, and we're happy to take on any questions.
All right, well, thank you so much. And we can move on now to the Q&A. As a reminder, you can still submit questions using the Q&A tab on the left side of your screen. I see we have a lot of great questions already. We'll try to get to as many as possible. To begin, what are the implications of changing the scale of production between early and late stage development? What about after PCS?
I can start with that question. So these things happen. Maybe the commercial need changes, and the volume of the product that needs to be produced changes. What I would suggest is having that life cycle in mind early on. It doesn't have to be too early. Let's say you're moving from CMC1 to CMC2. During CMC2, which is supplying for phase 2, phase 3 material, it would be great to have that final scale in mind. It's very easy to change scales during that time than after PCS. Not to say it cannot be done. It can be done, but it'll come at a cost. It'll come at a cost of delaying the timelines. So I would suggest do the scaling appropriately early on in the process.
Yeah, as Mini said, so as early as possible, because if you go into the scale down model development and PCS, you need a set of statistics significant runs in the commercial scale to really develop your scaled out model. If you don't have that, then you maybe need a intermediate scale or a pilot scale. And you can do that, but then you need to do some bridging studies at some point. So the earlier, the better.
Okay. Moving on, our next question. What criteria do you apply to determine FIT implementation through drug development from clinical development to commercialization? For example, cell culture media filtration to BDS filtration.
Okay, let me read this question.
I'll give you a second to take a look at the question, the text of the question.
Actually, I'm not sure what FIT implementation means. So maybe we can do that question a little bit later. Or Mini, do you know?
Yeah, I think we can address that a little bit later. I think maybe what the listener wants to know is how do we do the gap assessment? How do we do the facility fit from clinical development to
Oh.
commercialization? So at least from the downstream perspective, we are looking at scalability of our columns. We're looking at hold times. How fast can the product move through? What is the cadence? What is our planned schedule? But we can circle back on that question.
Yeah, fit is written in capital letters, so that if it's a facility fit, of course, you always, if you develop a process, need to look in your commercial facility because you develop a process that it fits in there. Yeah, that is always what we do and what you should keep in mind if you do the development of a process.
I am seeing a note that says that was intended to be facility fit. So,
Let's see. Okay, good.
All right, next question. What are some of the issues that you've seen in the past that may be learning points for GMP manufacturing?
So I can start with the downstream.
Yeah,
oh, go ahead.
So of course there are learning points during the development of a process, and every project is new, and because of that we send people into our manufacturing facility from the development. So we basically have experts who are supporting with the new product in our facility, people in our manufacturing facility on the floor, because they developed the process and they are the experts.
From a downstream perspective, I think the biggest issues are some very nuanced nuanced activities that don't really scale. So, for example, I can think of an example where your pH range is really narrow. That is really hard to achieve when you're dealing with thousands of liters of material versus just a few milliliters in our non-GMP area. So we do, because we work so closely with our GMP team, both on the MSAT and the manufacturing teams, we pay a lot of attention to what doesn't work in the manufacturing area, and we bring that back to our development. So our philosophy is we don't want to try to force fit a process into our manufacturing, but then what makes manufacturing robust? What are the pitfalls? How can we avoid deviations by developing a process that fits well into our facility?
All right. And circling back to the question we were discussing before, the client has clarified that by fit, they mean filter integrity test. So does that change your answers, your thoughts on that one?
So
Okay,
I can start with the filter integrity test from a downstream perspective, right? So there are a couple of places where we do the filter integrity test, which is on the virus filter as well as the final sterile filtration going into the drug substance. For sure, we check how filterability is in small scale. I mentioned already the refiltration. So for commercialization, this becomes critical where we are uh, for the BDS refiltration, whether it's going into a cryo vessel or a small bottle or a bag. This is a very critical operation, and I can follow up with the person asking the question if you need more information.
Yes, filter integrity test for upstream in our manufacturing facilities, of course, for the filter that are used for the media preparation in development, we usually don't do facility filter integrity tests.
Okay. Moving on, our next question, how can PCS be accelerated?
So yeah, one thing is the earlier you learn about the process, the better. So as I said, during the CMC2 development, look already into the robustness and in-depth understanding of your PCS. So the more you know about your process, this reduce the factors that are come out of the FMEA. Additional thing is you can implement engineering controls into the manufacturing facility to reduce overall your risk as well. And that is how you need to investigate less factors.
Yeah, from a downstream perspective, more data the better. But in many cases, you will not have enough data. So that understanding of those critical quality attributes is very critical. So to accelerate PCS, again, having that life cycle end goal in mind, you can already start eliminating some of the risk factors that are very low. And you can do non-PCS studies ahead of time, maybe during development with PCS in mind. And that way your risk ranking and the risk scoring becomes low. Having statistics can accelerate PCS. That way, you don't go into this massive experimental setup. We use statistics software to guide our DOE, and that can narrow down the parameters that need to be tested and the experiments that have to be done. PCS can be accelerated having qualified methods already in place. That is, sometimes when you're in this fast path, maybe your methods are not qualified, or maybe they're in the process of qualification. So that can add to the delay in testing the samples, which can, of course, lead to completion of PCS. So there are multiple strategies based on how much data are available. We have worked with programs where we just have one GMP run. How do you design an experimental setup for process characterization with N of one? And that's when you leverage your development data or some prior knowledge or literature knowledge to design um, a good accelerated PCS plan.
All right. The next question here. Do you use process analytical tools to monitor and optimize the process? If yes, what kind of PAT is implemented in upstream and downstream processes?
So we are working on different PIT tools in upstream, for example, biocapacitance probes to measure the viable cell concentration and viability, working on other different options. But this is mainly what we do in process development. They are not implemented in our manufacturing facility. But of course, if we identify a technology that is really important for a project or for a process, then we would also implement it later in our manufacturing facility.
From the downstream perspective, we are looking at again the most unmonitored step is our TFF. So we are looking at, for example, monitoring UV or monitoring pH, monitoring the Donan effect, which can really affect the stability of the processes. So we are actively looking at TFF, for example, but all others are currently standard monitoring of UV concentration, conductivity, and for clinical stages, these are adequate. And it is true that PAT is transforming the way we are, say, for example, batch disposition? Can we do many of those studies on the floor, the QC testing on the floor, and have just send out bioburden sterility out? So that is a question all manufacturing sites are looking into, and we are doing that as well.
All right. How many PPQ runs are required by the health authorities?
I can take that question. So the short answer is there is no guideline. But the long answer is you have to justify how many you chose. So there is a requirement that all BLA filing have to be supported by 3 successful PPQ runs, and they have to be consecutive. So you can choose as a sponsor or as a CMO, you could choose to run as many PPQ runs as possible. You have to demonstrate 3 consecutive runs. And that, again, that confidence comes from how much data you have during the clinical phases. If you've had a lot of runs and if you have not changed the scale, your processes are robust and you have consistent data, you could just be aggressive and just plan for 3 PPQ runs. But if there are any doubts and if there are major process changes that have happened and you don't have that clinical confidence, you may want to plan for, say, 5 or 7, and then you can also stagger them. So there's one strategy where you could run 3 PPQs in a row, and you pause and you assess. And if those runs meet all criteria and they satisfy all validation requirements, you are done. It's great, but you always should have that backup plan, and maybe not spread them out too much. Again, that could contribute to delays in filing the BLA.
Okay, well, thank you. We are coming to the end of our hour, so this will have to be our final question. We did have a lot of great questions today. We couldn't get to them all, unfortunately. We will do our best to get back to everyone after the webinar to address any questions that haven't been answered. For the final question, what are the regulatory commitments after BLA and during commercial manufacturing?
I think I mentioned that a little bit in terms of establishing the CPV program, and then you have your inspection commitment. So when you file the BLA, the FDA, as an example, has a certain number of days that they have to get back to you. So during that time, you have to have your quality and regulatory teams ready for answering any such questions. And then the response time is very short. So you have to have robust strategies as to how you would answer those questions. Feeding them all the information is not a good idea. Feeding them less information is also not a good idea. So again, it requires careful planning how you would address those questions. The other commitment you have to the agency is to have a robust training plan for all the operators and the folks involved. And so you have to start getting ready for that pre-approval inspection, and that is part of your commercial readiness. And of course, you're continuing to manufacture commercial batches. You may be doing that already. So it's not enough that you've proven through the PPQ that you can make those commercial batches, but through this process of filing the BLA, if you're continuing to manufacture commercial batches, you should be doing them with all of the regulatory requirements in mind.
All right, well, thank you so much. And I'd like to thank everyone for attending this Fierce Biotech webinar and for submitting all these great questions. I'd like to thank our speakers again for participating and BioNova Scientific for presenting today's webinar. A recorded version of this webinar will be available for you to access within 24 hours using the same audience link that was sent to you earlier. Thank you again for joining us, and we look forward to seeing you at future events.